Okay, so welcome to this next video in which we are discussing the adrenoceptors. Okay, right. So we've now discussed G-protein coupled receptors, we've discussed the G-protein cycle. What I now want to discuss is the diversity of heterotrimeric G-proteins, because there are a huge number of different heterotrimeric G-proteins, which is because there is a huge number of different alpha subunits for heterotrimeric G-proteins, there is a huge number of different beta subunits, and there is also a huge number of different gamma subunits, okay? So there's a massive number of heterotrimeric G proteins that you could potentially make. Okay, so let's start off with the diversity in the alpha subunits. So basically there are 16 genes for alpha subunits, okay, and these produce 21 different known subunits for alpha subunits of heterotrimeric G proteins. Now you might think, well, what? That's an anomaly. How can we have 16 genes producing 21 different proteins? Well, basically it's because some of these genes have different splice variants. So let me just remind you of what that means. So let's recite our central dogma of biology. Okay, so we start off with our DNA here, and let's say we have some gene for an alpha subunit of a heterotrimeric G protein. So we take one of these genes, okay? Now, basically, when RNA polymerase II works its way along the DNA, producing a piece of mRNA for that gene, it will produce a piece of mRNA that is complementary to one of the strands of the DNA. Okay, so actually it only needs one of the strands of the DNA, not both. Now the strand that it actually uses to make a piece of complementary mRNA is called the coding strand. So let me highlight this in pink. So let's let this one in pink represent the coding strand of the DNA. So the strand which RNA polymerase II is actually going to use and make a piece of mRNA that is complementary to. Okay, the other strand which is complementary to the coding strand and is shown here in turquoise, this is called the non-coding strand. Okay, and RNA polymerase II doesn't actually need that. Okay, right. So what's going to happen is that RNA polymerase II will work its way along the coding strand and produce a piece of mRNA that is complementary to the coding strand. Okay, so I've shown that here, and I'll colour it in blue. So this piece in blue is the mRNA, and this piece of mRNA is not the thing that is then going to be translated. This piece of mRNA that is directly complementary to the coding strand, which I'll colour in here in blue, this is known as the pre-mRNA, or the primary transcript. Okay? And this is not the piece that will actually go through the ribosome. If you put this straight through a ribosome to be translated, you would get a protein that was just nonsense, basically. Okay? And the reason is that not all of this mRNA here is actually meant to be translated. Okay, so there are some portions that you need to translate, i.e. there are some portions that actually should be read by ribosome and converted into protein and will make a part of the protein for which this gene codes. So let's show these here. Now these portions that should be included, these are called the exons, okay? So these three here, these are exons. The portions of mRNA that are in between the exons, which are sometimes called junk coding sequences, these are called the introns. Okay, so these are the introns here. Now, basically, what has to follow is a process called splicing, where basically you cut the introns out of the mRNA, and then you stitch the exons back together to make a smaller piece of mRNA that can then actually go through a ribosome. Okay, and this smaller piece of mRNA that is actually ready for translation, this is then called a mature mRNA. So this is the mature mRNA. Right, and this will then be translated into protein. Now, the reality is that splicing doesn't always, well, not in every case, does splicing actually always produce exactly the same mature mRNAs. So in some pre-mRNAs, there are different ways that you can splice the pre-mRNA, and you can end up with different mature mRNAs. So for instance, some pre-mRNAs might have exons that are actually optional. Now this is quite an extreme example, but some pre-mRNAs will have exons that you don't actually have to include. You can include them if you want, but you don't have to include them. Okay, so when you make the mature mRNA, you can either have this exon or you don't have to have this exon, and then the two pieces of mature mRNA that you then get will produce different proteins. The one with the extra exon will produce a slightly longer protein than the one without it. Okay, and both of those proteins might well be functional, and then you've produced two separate proteins basically from the same gene. That's quite an extreme example. More often, it's just a case that you can choose. For certain exons, it's variable how much of that exon you actually include, so it might not be necessary to include the entire exon. You might only need to include half of it or something along those lines. So for some of the exons, there will be variable levels of inclusion that you can have. So the point is that from one type of pre-mRNA, you can make a great number of mature mRNAs in some case, and all of these different mature mRNAs will then produce slightly different proteins, and these different proteins are then called the different splice variants of this gene, basically. So some of these genes will produce different splice variants, and that's how, even though we've only got 16 genes, there are 21 different known alpha subunits. Okay, right. So, let's now discuss these genes then. Okay, so we're not going to go into detail with the different splice variants, but we are going to discuss the 16 different genes for alpha subunits. Okay, now to make it more easy to comprehend, the alpha subunits are grouped into 4 families. Okay, so the first family of alpha subunits of heterotrimeric G proteins is called the G-alpha S family, and this contains 2 members. It contains the G-alpha S gene itself, and then it also contains the G-alpha Olf gene. Okay, and both of these are very, very similar. G-alpha Olf is actually really just a version of G-alpha S. It is a separate gene, but basically G-alpha Olf is the alpha subunit which replaces G-alpha S in certain tissues of the body, for instance the olfactory epithelium, which is why it's called G-alpha Olf. So in certain places in the body, instead of having G-alpha S expressed, you'll have G-alpha Olf expressed, but they do pretty much the same thing, which is why they're grouped together in a family. Now, this family also allows us to explore the concept of different splice variants, because the G-alpha S alpha subunit has a huge number of different splice variants, whilst G-alpha Olf only has one protein that is known that it can produce. Okay, but we won't go into the different splice variants of G-alpha S. Okay, next family. The next family is rather bigger. Okay, it's the G-alpha I/0 family of alpha subunits of heterotrimeric G proteins, and this contains 8 members. Okay, so it contains firstly the G-alpha I1 gene, then it contains the G-alpha I2 gene, then it contains G-alpha I3, and these are all separate genes which code for separate proteins, so they are not just different splice variants of one gene. So you might have thought that there was just one G-alpha I subunit, but actually there are 3 of them, okay, and they're all coded for by different genes. And then there are the transducin alpha subunits, which are G-alpha T1 and G-alpha T2. Okay, so basically in the rod and cone cells of the retina, you have photoreceptors, receptors that respond to light. Rhodopsin is an example in rod cells. Okay, it's a slightly different variant in cone cells. Okay, but basically rhodopsin, when light hits it, it's a a G protein coupled receptor, and it will change conformation, and it will then be able to interact with a heterotrimeric G protein. And the heterotrimeric G protein that it interacts with is called transducin. Now, in rod cells, the alpha subunit of transducin is this G-alpha T1. So this is the alpha subunit of rod cell transducin. Okay, now in cone cells, not only do you have a slightly different photoreceptor, it's iodopsin rather than rhodopsin, but you also have a slightly different form of transducin. Okay, so the alpha subunit of the heterotrimeric G protein of cone transducin is G-alpha T2, so this is the alpha subunit in cone transducin. Okay, right, so let's go on then. So that's 5 of the members of this family. Then you also have the G-alpha O gene, okay, the other one after which the entire family is named. Then you have G-alpha Z, and then finally one that's involved in the gustatory system, or G-alpha gust, basically. Okay, so that's the 8 members of this G-alpha I/0 family. The one thing just to mention is that if you read the literature, you will often hear people talk about the 2 different splice variants of G-alpha O, and the terminology, well, the notation for these is quite nice. And also, I don't want you to think that they are separate genes, okay? So there are 2 separate splice variants that are known of G-alpha O, and these are G-alpha OA and also G-alpha OB. Okay, so those are the 2 separate splice variants of G-alpha O, and they're not separate genes, they are just the 2 separate splice variants of this one G-alpha O gene. Okay, so don't let that confuse you. And then we have the next family of alpha subunits of heterotrimeric G proteins, which is the G-alpha Q/11 family. Now, the most famous member of this family is the G-alpha Q alpha subunit. Then you also have G-alpha 11, then G-alpha 14, and then finally one that's called G-alpha 15/16. Now, I don't want you to think that this is 2 separate alpha subunits, okay? So I'm going to tell you the story behind why it's called G-alpha 15/16. So basically, long ago when they were discovering these, well, not too long ago actually, basically people working in mice found a new subunit. They found a new alpha subunit for heterotrimeric G proteins, and they called it G-alpha 15. Okay, then people working in the human found a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 16. Okay, later it transpired that actually this G-alpha 16 that we had found in humans was actually the same thing as we found in mice, which was called G-alpha 15. They were the species equivalents, basically. Okay, so there is now a movement to rename both of these G-alpha 15/16. However, if you're talking about mice, you will still find people commonly referring to G-alpha 15, and if you're working in humans, you will see people often referring to G-alpha 16. But basically, they're just the 2 different names for the same alpha subunit in 2 different species. Okay, so it won't be uncommon if you're working in the human to hear people call this just G-alpha 16 rather than G-alpha 15/16. Okay, then the next alpha subunit family is the G-alpha 12/13 family of alpha subunits, and this contains G-alpha 12 and then also G-alpha 13. And now I think that's all of our 16 genes, so let's just count up. So we've got 1, 2, 3, 4, 5, and this is 1, so 6, 7, 8, 9, remember this is 1, 10, 11, 12, 13, 14, and then the 2 up here, 15, 16. So that takes us up to 16 genes for alpha subunits. And as I say, we're not going to go through all the different splice variants of them. Okay, we've looked at the splice variants of G-alpha O, but we're not going to go through the splice variants, certainly of G-alpha S, because the notation for it is just hideous. Right, okay, so let's turn our attention to the beta subunits of heterotrimeric G proteins. Okay, so beta subunits. How many different genes are there for beta subunits? Well, basically there are 5 genes for beta subunits, and then there are 6 actual subunits that are known. Okay, now this might have shocked us about 20 minutes ago, but we understand this now. We know exactly what's happened here. One of these genes has produced 2 different splice variants. Okay, and that's exactly what's happened. So, basically the 5 different genes for beta subunits are called beta 1, beta 2, beta 3, beta 4, and beta 5. So nice, sensible naming. Okay, so those are the 5 different genes, and basically the beta 1 gene will produce... Oh, actually, I should just add in, they're called G beta 1. Strictly speaking, I should put a G there, okay, just to be specific. So you should put G, and then you should subscript beta 5. Okay, so this is G beta 1, G beta 2, G beta 3, G beta 4, and G beta 5. And G beta 1 gene codes for the G beta 1 subunit, the G beta 2 gene codes for the G beta 2 subunit, the G beta 3 gene codes for the G beta 3 subunit, the G beta 4 gene codes for the G beta 4 subunit, and then finally, G beta 5 is the one with 2 splice variants. So it produces the G beta 5 subunit, and then also the G beta 5L subunit. Okay, so it produces 2 different splice variants to give us overall 6 different subunits for beta subunits of heterotrimeric G proteins. Let's now turn our attention to gamma. Okay, so gamma's the simplest. Gamma has 12 genes, and there are also 12 subunits, which is nice. Okay, now the naming of the gamma subunits is also very simple, except there is one little blip, basically. Okay, so there is the G gamma 1 subunit, there is the G gamma 2 subunit, there is the G gamma 3 subunit. Okay, there is the G gamma 4 subunit, there is the G gamma 5 subunit, and then here's the blip. There is no G gamma 6, so we skip straight on to G gamma 7. And then we go on all the way up to 13. So G gamma 8, G gamma 9, G gamma 10, and then we'll go on to the next line, G gamma 11. And you might think, well, don't we have to stop at 12? But no, because we missed out 6. So we won't have enough if we stop at 12, so we need to go one more to G gamma 13. Okay, and that now produces us the 12 different gamma genes, which all code for one protein as far as we know, and therefore the 12 different gamma subunits. So, hopefully what that has impressed on you is the absolutely huge number of different heterotrimeric G proteins. Now, idyllically, if you were going to name a heterotrimeric G protein, you would tell me exactly which alpha subunit you have, exactly which beta subunit you have, and exactly which gamma subunit you have. For example, if I wanted to name a heterotrimeric G protein, I could name it G alpha I1, that tells me that the alpha subunit is I1. Then I could have beta 1, that would tell me that the beta subunit was the G beta 1 subunit, and the gamma subunit can also be gamma 1. Okay, so that would completely specify a heterotrimeric G protein. However, people very rarely do this, and the reason is people very rarely know the exact subunit composition of the heterotrimeric G protein they're working with. Especially, people do not generally know the beta-gamma subunit, so people never really ever tell you what the beta-gamma subunits specifically are of the heterotrimeric G protein, and usually G proteins are just named after the alpha subunit they have. Okay, so you might see this called GI1. Okay, and even that is them being very, very good to you, because they are telling you specifically which alpha subunit you are dealing with here. Generally, people don't even know specifically which alpha subunit they're dealing with, especially when they're dealing with this big family of G alpha I naught. So often, people would call this just GI, and what that would tell you is that you are dealing with one of these three here, i.e. the alpha subunit is one of the I subunits, okay? even more common, people don't actually even know that, and they would just say that this is a GI/naught. So they would tell you that it's in the that its alpha subunit is within the G alpha I/naught family, but we can't really tell you much more than that, basically. Okay, so that is the naming of heterotrimeric G proteins. So often you will see heterotrimeric G proteins named just by their alpha subunit, and often we don't even know specifically which alpha subunit it is. So we'll either give you a sort of clue as to where it is. So we'll tell you, for instance, GI would tell you that it's one of those 3I ones that we've seen. GT would tell you that it's one of the transducin ones. GS is quite nice. Generally, if people put GS, that means that the alpha subunit is an alpha S. But again, there's loads of splice variants of alpha S, so it doesn't tell you specifically which one of those it is. Okay, but then even further, going even further backwards, some people will just tell you which of the 4 families the alpha subunit is in. Okay, so this tells us that the alpha subunit is in the G alpha I/naught family. Okay, right, and that's simply because people generally don't know exactly which alpha subunit, exactly which beta subunit, and exactly which gamma subunit they are dealing with. Very, very rarely will people ever tell you what beta and gamma subunit you're dealing with, because we don't know, and it doesn't seem at the moment as though it matters terribly, okay, which specific beta and which specific gamma subunit you have. Generally, we seem to, at the moment, at least as far as we know, it seems that most of the beta-gamma subunits, no matter what the beta and gamma subunits specifically are, kind of do the same thing, basically. But of course that's probably very naive, and it will probably change in years to come. Okay, right, so we'll call it there for this video, and in the next video what we'll do is move on to discuss the alpha-1 subunit in more detail. Sorry, not the alpha-1 subunit, the alpha-1 adrenoreceptor in more detail.